Uh-oh. Okay. All right, good. Okay, we're going to continue with the podcast. No, actually, this is the new podcast for you people at home. All right, any rate, because you don't know how to do G2 and M, okay? You can't distinguish it with the flow cytometry. So the question is, well, how do you determine mitosis? And you can determine mitosis because you can actually see what's happening in mitosis. So the first thing that happens is the nucleoli, which is where ribosomes are being assembled, before anything else happens, that disappears. So you can just see the nucleoli disappearing inside the nucleus. And that's visible with a light microscope. Then the nuclear envelope, it's easy to see the nucleus. Nuclear envelope breaks down, okay? And then you get a mitotic spindle forming, and eventually you get cytokinesis. So you can follow these things, and you can follow the chromosomes condensing during mitosis through what's called a karyotype. So there's a number of things that you can follow. Okay, so now we're gonna start looking at how the cell cycle has been analyzed, and there's three major approaches that were used initially. First one is genetic studies in yeast, cell cycle mutants. Then there's xenopus oocyte maturation, which is a type of biochemical study. And then sea urchin oocytes, which is going to be a more cell biological study. All right, so we're going to start off with these first, looking at the cell cycle mutants in yeast. Okay, so first we have to be familiar with the cell cycle in budding yeast. Okay, so here, what you see is that in G1, you have a small round cell. And as it makes a commitment at start to enter S phase, it's a much larger round cell. And then you can tell S phase because it starts making a bud. So these are budding yeast, and the bud only starts in S phase. So as soon as you see a bud, you know you're in S phase. The bud gets larger, and then eventually in mitosis, what will happen is the nucleus is going to go from the mother bud, the mother cell, to the daughter bud. And in budding yeast, there is no nuclear envelope breakdown. Okay, so it's going to go in between and then segregate the DNA within the nucleus. And so what you can see here, there's actually microtubules forming inside the nucleus in budding yeast. And then cytokinesis will occur, cutting the cell in half, and you end up with a mother cell that's larger and a daughter cell that's smaller. Okay, so this is the thing. And so the main point is that you can tell where you are in the cell cycle based on the size. And the shape. Okay, and as I mentioned, start is where is a point in the cell cycle that the yeast are going to determine if they're going to go into the rest of the cell cycle or not. And they're looking at certain things. They're looking at, is there enough food here? Enough food to go in? Have I grown large enough? Because the cell has to get large enough before it goes in, because this is actually the growth phase. So if a cell didn't grow, and it just started dividing, it would just keep getting smaller and smaller. So clearly the cell has to get big enough so when it cuts itself in half that you end up with two good-sized daughters. And then these yeast are haploid, and they can mate. Okay, so if conditions are not good, there's not that many food, and then they're saying, oh, I want to mate, and because conditions are not that good. So if they decide to mate, then they're not going to go through the cell cycle either. Okay, and the mating is so that they'll produce spores. Okay, so conditions are not good. What they're going to do is they're going to go through meiosis, and they'll form 4 spores. And spores are pretty much resistant to everything. They're basically like a seed that is just waiting for good conditions before it turns into a functional yeast. Okay. So this is what the yeast is looking for at start, and if it gets a good signal on all of these, it'll go through the cell cycle, and once it passes start, it's committed to the cell cycle. Unless you do something to the yeast, like you insult it with some kind of a chemical to stop DNA replication, or just something that is not normal, once it makes the commitment, it's going to go through the entire cell cycle. Okay, so this is the commitment point at start. Okay, so that's one system that's used to look at these cell cycles. The other one's fission yeast. So fission yeast has a different shape, and it's got this sort of oblong shape, and you can tell where it is in the cell cycle just based on its size. So as it's going from S phase, it starts off as a pretty small oblong cell, and then as it just keeps getting larger. But unlike budding yeast or mammals, G2 is the growth phase. Okay, so G2 is where it gets bigger, and then it's going to form. It's called a septum, like a cell wall in mitosis. And actually in G1, it's still connected. Okay, so you have two cells that are connected. They have a cell wall, so they've gone through mitosis, but they're still connected. So you can tell G1 because you got these cells connected, two cells connected, and in S phase you can tell because they're really small. Okay, and then in G2 you can tell they're there because they become really big. Okay, so you can tell where you are in the cell cycle with fission yeast also. Okay, so basically the screen that they're going to be using is using the shape of the yeast as a readout for where they are. And as we just went through this, you can tell where they are based on that. This drawing's not correct for G1 and fission yeast because, in fact, remember, they're actually still connected in G1. Okay, so here we have, this is the basic part of the screen. You've got cells that are going around the whole cell cycle at the permissive temperature. And okay, so first off, they're going to use a temperature-sensitive mutants. They're going to first screen for temperature-sensitive mutants, and then they're going to look for mutants that at the high temperature where the protein doesn't work anymore, they're going to arrest at one point in the cell cycle. So if you had one of these temperature-sensitive mutants that was affected the cell cycle, then you shift it to the high temperature. What's going to happen? The cell in S phase is going to go through G2 and M, come around and get stuck in G1. The cells in G2 is going to go through mitosis, come around and get stuck in G1. Cells in mitosis going to go through and come around and get stuck in G1. And the cells in G1 is going to get stuck in G1. Okay? So you can just look and say, oh, whoa, everybody's at G1. So it's a cell division cycle mutation. Okay? All right, so this is how you would do the screen. So initially, because you're looking for temperature-sensitive mutants, that's going to be, most mutations are not going to be temperature-sensitive, so you're going to mutate the yeast randomly, and then first get your hands on all the temperature-sensitive mutations. And then once you have those, you're going to test to see if they're cell division cycle mutants. So what would be a reason why you'd want to first, get temperature-sensitive mutants? Why would you want to do that? Why would that be important? Mm-hmm. So you're saying so you can determine when these, that's true. It'd be very useful to be able to say, okay, I'm gonna be able to control it. And that's true that it's a super useful tool. But why is it actually more important than that? Why is it more important than that to have temperature-sensitive mutants? Somebody say something? All right, and let me just put it this way. It just has to be conditional. It has to be so you can turn it on and off. Why would you need to make it conditional? What would happen if a cell division cycle mutant were not conditional? Yeah.
Would they just would have any control over it? They could continue like running or going the cell cycle.
So what would happen if it wasn't conditional and it was required, let's say, to go from G1 to S phase? Well, let's say it wasn't conditional. I mean, let's say you're hitting these genes, right? So you're hitting a ton of genes, and you hit a gene that's required to go from G1 to S phase. What's going to happen to that one cell? It can't go from G1 to S phase. Huh? What'd you say? It's going to keep growing? Yeah, it's not going to keep growing, right? What's that? Yeah. Well, it's going to get stuck at G1, right? It's going to get stuck at G1, and it's going to die, and it's just a single cell, right? So if you want to be able to grow up a whole mess of these things and keep it going, you have to make it conditional. Because I mean, if the cell can't get through the cell cycle, it's going to arrest at that point. So first you have to make it conditional, and the easiest way to make it conditional is just temperature sensitive. So they first have, here they've done a replica plate of these colonies that are mutagenized. Each individual yeast is forming a colony. They replica plated. They grow at 2 temperatures. They say, okay, well, this colony is growing at 25 and it doesn't grow at 35, therefore it's temperature sensitive. Okay, temperature sensitive for growth. But you could have temperature sensitive for all kinds of things. It could be like, oh, it's needed to make ATP. It's some kind of metabolism enzyme. It's required for, homeostasis. Who knows what it is, right? A lot of those things are not going to affect the cell cycle. So then you take this temperature sensitive colony, you grow it up. It's actively growing. And then you shift it to the non-permissive temperature and you ask, when they stop growing, okay, because clearly they can't grow at the high temperature, right? When they stop growing, are they spread out among the whole cell cycle, or is it all at one point? If it's at one point, they're going to say it's a cell division cycle mutant, must have something to do with the cell cycle. So that's the rationale. Okay, so then if you're working in yeast, there's a super quick way to figure out what that mutation is. Okay, so you've got a mutation in one gene that's making them stop when you shift it to the high temperature. And you're pretty sure it's one gene because it's temperature sensitive. Okay, temperature sensitive, not easy to get. And so you probably only have it in one gene and you shift it to the high temperature, they stop growing. Therefore, at the high temperature, that gene's activity is missing. So because the yeast genome is very small, you can just chop it up into pieces, throw it into a vector, and then put it into the yeast that are the CDC mutants. And so when you shift to the high temperature and you put in just a random gene like gene X, nothing happens. It still arrests, okay? But if you were to put in the actual gene that's mutated, like in this case, it's cell division cycle gene 28. So it's CDC28 mutant. Doesn't have CDC28 at the high temperature. You put in the wild type gene, well, now it's going to work, right? The wild type gene is not mutated, okay? So then you get the colony growing up, and then you just isolate the plasmid out of it, and there you go. You can just sequence a plasmid, and you can figure out what that gene is, okay? So this whole thing only takes about 2 weeks to do. It's called a complementation screen, where this wild type gene is complementing the mutation. So now it's no longer arresting. All right, so Paul Nurse did the screen in fission yeast, and he got one mutant, CDC2, that arrested at both G1S and G2M cell cycle transitions. And then Lee Hardwell was doing the screen in budding yeast, and he got the ortholog. So the ortholog means it's the same gene in a different species, okay? So it's the counterpart. This is budding yeast CDC28 and fission yeast CDC2, and they both got it in their screen, okay? So one, it's conserved in the two yeast. And that's sort of important because the yeast are separated from each other. Budding yeast and fission yeast, they branched really early, and they're separated from each other pretty much the same genetic distance as against vertebrates. So they're really distinct, okay? And so, we're dealing with like about over 1 billion years of evolution distance between budding yeast and fission yeast. And so you got the same one. And his also rested at G1S and G2M. So you got the same gene, and it's required for G1S and G2M in the two things. Okay, so Paul Nurse was super interested in CDC2, and you could ask yourself, well, why would Paul Nurse be interested in CDC2? And the rationale is, I just told you, so here I'm going to the overhead thing. Okay, so you can have an arrest, G1S, and they got an arrest, G2M. All the cells accumulate here, and they're accumulating here. Okay? I just told you that the screen was for arresting at one point in the cell cycle. So the question is, well, how is this thing useful? Right? We're arrested in 2 points. It seems less specific. Like, why would he be interested in this one? But this is the one he was super interested in. He was way more interested in this than anything else. So he wanted to get the human counterpart. But why would he be interested in this one, but not another CDC? Because he's got a ton of the CDCs that arrest at this point or at this point, but just 1 point. It's the only one that arrested at 2 points. So why would arrested at 2 points be useful? So if you're going to answer, raise your hand so I can see where you're at. So the answer is more opportunities for a drug target. No. Nope. Yeah.
Would it be some kind of variant?
Some kind of variant. Well, I mean, that's what it's normal. It's needed for those two transitions, right? But so these are all different genes. So why would this gene be needed for two transitions, but the other one is only needed for they're only needed for one? Well, let me put it another way. Why is it sort of important that? What's the advantage that it's required for two transitions, or what does that tell you? Yeah.
You can tell where the mutants were before they got arrested.
Well, they're all going to rest somewhere. Like you're going to have a whole mess of mutants that arrest at G1 to S. A lot are going to rest in G2 to M. Some are going to rest in S phase. You're going to have other ones arrested in mitosis, right? So you've got all these different kinds. With a what? You said checkpoint? The checkpoints, so we're going to get to checkpoints later, but checkpoints are, let's say you hurt the cell in some way, like you caused DNA damage. The cell's going to be like, whoa, I gotta stop and fix it, right? That's what a checkpoint is. But these yeast, we're not actually doing anything except inactivating this gene. We're not actually activating we're not causing DNA damage or something that the cell would respond to with a checkpoint. Yeah. Redundancy. So redundancy means that they're going to be like different genes doing the same thing. But if they were redundant, then you wouldn't even get it, okay? Because you need this gene to do G1S, but another gene can do the same thing as this gene. And you need to take this one, the other one will just do it. So redundancy is opposite of this. Yeah.
Because it's a single regulator that's controlling, that's right before two of the sort of more you can't stop phases like DNA replication and then mitosis.
So I think that you're very close. What are you saying again?
Because it's a single mechanism that stops, if it's mutated, it stops cells right before two of the, I guess not really most important, but more transformative phases.
Well, okay, so I think that's pretty much sort of on track. So basically, if the cell's arrested between G1 to S, okay, it can't do S phase. In S phase, there's a whole mess of things that you could mutate in S phase, like polymerase cell, proliferating cell nuclear antigen, DNA binding elements, who knows, there's a ton, right? Okay, mitosis. Well, you've got these huge rearrangements of the cell. You've got, you're rearranging the cytoskeleton, you're doing all these, breaking down nuclear envelope, all kinds of things, right? A lot of things you need for mitosis, right? Well, if you're arrested at G1 to S, means you're affecting, either you're affecting something about S phase, and you're affected G2 to M, maybe you're affecting something in mitosis, or you're the regulator. You're determining, this protein's determining if you're going to S phase. This protein's determining if you're going to mitosis, because it's going to be unlikely that you need some biochemical activity for both of these things. It's more likely this is the regulator. This is the protein that's determining if you're actually going to do the transition, okay? So this looks like it's actually controlling the transitions, and it's controlling two of them, okay? That's why he's interested in it. He wants to know what the regulation of the transition is. Okay, so this is the one that he wanted to get his hands on, the human one. So people in the human field who were studying the human cell cycle, they had no clue what was going on. They didn't know how it was regulated. And they weren't that interested in yeast work, because they were thinking, oh, well, yeast is like a billion years separated from us. It's probably a totally different mechanism. So he said, okay, I'm gonna clone CDC2 by complementation for the human gene. So what he did was he said, I'm going to make an assumption that the human genes also work in the exact same way. And if I have a mutant that doesn't have, in this case, CDC2 or CDC28, same gene, I can put in the human gene and it will work instead of the yeast gene. And the yeast can't divide because it's lacking that gene. If I put in the human gene, it'll divide. So this is pretty outrageous. He's saying that over a billion years of evolution, the gene will still work in the other species. And so he put in a human cDNA library and to be expressed in yeast, and sure enough, he comes up with something that allows him to grow. He sequences it, and it looks just like the yeast gene. So he's got the ortholog in human. And what that tells you is that the human gene is now also working in the same way. It can take the place of the yeast gene. Okay? Suddenly, all the people who are studying humans were like, whoa, that means that almost every CDC gene is probably going to be something important. And they've got a whole mess of them. They've got a lot of CDC genes. So suddenly everybody started paying attention to what the yeast people were doing because they realized that over this billion years of evolution, how the cell cycle is controlled is actually conserved. Okay, so we're going to switch to what the vertebrate people were doing at the same time. They were looking at Xenopus, which is a frog, African green frog, and they're looking at oocyte maturation. What time is the class end?
4:45.
Okay. So they're going to be looking at meiosis in the oocyte. Okay, just to remind you what's happening in meiosis. So the cell is 2N. It's going to replicate its DNA. It's called pre-meiotic S phase. So now it's 4N. Okay, okay, what does this mean, 2N? So that means, let's say we're looking at one chromosome. Let's say we're looking at chromosome 1, and one of the chromosomes, the red one, let's say you got it from your father. And then the blue one, you got it from your mother. So you have 2 copies, right? One of them came from the oocyte, that's from your mother. One came from the sperm, that's your father. So you end up with 2 copies. And those copies are going to duplicate, okay, in the pre-meiotic S phase. Then meiosis 1, the 2 homologs, so that's chromosome 1 from the father and chromosome 1 from the mother, are going to pair up. And there would have been some recombination earlier. And then they're going to segregate. So now you end up with one copy, with just one of the chromosome homologs. There's a little bit of recombination, but it's basically either got the father copy or the mother copy. And then in meiosis 2, you're going to go through and segregate the 2 copies that you copied them here in the S phase. You're going to segregate them away, so you end up with 1N. Okay? So you start off with 1 diploid cell, you go through S phase, and you end up with 4 haploid cells. Okay? So let's take a look at what's happening in oocyte maturation. In oocyte maturation, they're like in the interface before this whole thing, and then they're going to go, they get a hormonal trigger, and then they're going to go through meiosis 1, which is this whole process here, okay? And then they're going to dump off one of the DNAs. So one of these DNAs gets dumped off in a polar body, so it doesn't continue. And then they actually arrest here with the meiotic spindle set up, waiting, and they're going to hang out here until they get fertilized. When they get fertilized, they're going to dump off the other DNA. They're going to dump off this DNA. Okay? And so that's it. But maturation is just going from interface to this first meiosis, and that's a hormonal trigger. And we're going to be focusing on this thing, which is basically going from an interface position to meiosis. Okay? And but before we do that, let's take a little side step and figure out why this is actually happening. Why are they throwing up polar bodies? Basically, they're going through here, and they threw off half the DNA here, and they're only left with half. This is all gone now. And then they're only left with half the DNA, and they threw out that half too. So it turns out females in all these animal species, including every female here, are going to go through this thing in meiosis. So while the males are all going to, like when the males go through meiosis, they end up with 4 sperm. But when the females go through this meiosis process, they end up with 1 oocyte. And they dumped out all the DNA, and they basically got rid of it. Why would females do that? What's the rationale of females? What would be the advantage? It seems like you're throwing away 3/4 of the DNA. Doesn't seem smart to me. What would be the rationale? So let's look at this in the context. You're basically throwing away the little bit of DNA with very little cytoplasm here, and the DNA with a very little cytoplasm here. So what would be the rationale for doing that? So we're just talking about a biological explanation. I mean, men can answer too. Go ahead. Yeah, that's right. So basically, this oocyte is huge. It's a huge oocyte. And it's loaded with mRNA and protein and all these things for those rapid cleavage divisions we talked about earlier, right? Like as soon as this thing starts going, they're going to start dividing in super fast ways. It's only capable of doing that because they've been loaded up with everything they need, histones, polymerases, everything, okay? Well, it's a lot of effort to create all of that cytoplasm that's loaded up with what's called maternal product. And so it's not in a female's interest to split it into 4 pieces. Then they have to grow up all 4 of those pieces. It's just way easier because the rate limiting step is growing the oocyte to just dump off the excess DNA. So you preserve that oocyte for these cleavage type of divisions that are going to come. All right. So, but what we're focusing on now, so that was a sideshow. What we're going to focus on now is maturation, where they go from interphase to this first, arrested meiosis 2. Okay, so let's say you take this cell that's arrested in meiosis 2. It's mature, okay? And it's waiting for the sperm to come in. You take some of the cytoplasm and you put it into an immature oocyte, it will mature, okay? So there's something in this cytoplasm that will promote meiosis or promote maturation. So that's why it's called maturation promoting factor. If you take extract from an interphase cell, put it in an interphase, nothing happens, okay? So there's some activity that's in this that will allow an immature oocyte to mature, okay? So that's why it's called maturation promoting factor. And because we're talking about like basically going into like a mitosis-like state, we're talking about this, it's basically a G2 to M transition, but it's in meiosis. But it turns out that it's not just meiosis. You can also take interphase from a mitotic cell, not meiosis, mitosis. And it'll also cause it to mature, okay? So whatever this factor is, it's the same. Whether you're making the cell go into meiosis or into mitosis, it's the same thing. And then you can see this mitosis effect when you do cell fusion assays. So like if you take a mitotic cell and a cell in G2, you can like add these long anions that are going to go in the membrane and cause the membranes to fuse together. And then when they fuse, suddenly you have a nucleus from the G2, this is G1, the G1 cell with a mitotic cell. And what will happen is immediately that G1 phase nucleus is going to go in mitosis. And when it goes in mitosis, it's going to get nuclear envelope breakdown, and the DNA is going to condense. But you can see that, like these are these small mitotic chromosomes. You can see that, like here, but you can see that these chromosomes are long. So if you force a G1 nucleus to condense its DNA, it's not that condensed. If you combine a mitotic cell and an S-phase cell, the S-phase nucleus is immediately going to go into mitosis, but it's in the process of replicating its DNA. So you're going to get this kind of shredded appearance because you get all these cells that are in the process of doing replication forks and things, right? So you force it to condense, it's going to look like this. And then if you take a G2 cell, well, it'll go into mitosis prematurely too. But what you see is that the G2 cell looks like here's the super small mitotic chromosomes, and like here's one G2, and you can see that it's way bigger. So, but it's better than the G1 one, right? The G1 looks like this. So what that tells you is that even in G2, they've already started to condense their DNA a little bit, okay? And you don't get the full condensation until mitosis. But I mean, if you force it, it'll condense, but it won't be as good as it is in mitosis. At any rate, what this is demonstrating is that there's some factor in the mitotic cell that's forcing the other cells to go into mitosis. Same thing with the maturation promoting factor, okay? But they have no clue what it is. All right, so they're stuck right there. They don't know what it is. And now the last one is sea urchin oocytes. This guy, Tim Hunt, did this. And so his idea was, I'm going to see what is expressed in the early cycles. And he's going to use sea urchin. And so his approach is to label with S35 methionine. And you know that all proteins start off with methionine. So if you label with S35, every protein is going to have at least one methionine, and it's going to get radioactive. Okay, so it's radioactive methionine. And then he says, okay, I'm gonna run the whole extract on the thing, and I'm gonna see what's being made. So what you see is that there's not a lot of synthesis. So he's gonna fertilize them, and then he's gonna say what happens after they're fertilized, start going through mitosis. And what you see is that there's not a lot of synthesis here. You got this thing, it's called cyclin A, this one's cyclin B, and you got ribonucleotide reductase. And this one just keeps going up, but these ones cycle. So let's step back for a minute before we think about what this means, and just ask. You can always ask yourself. Scientists aren't like crazy generally, right? And you're like, sea urchin, huh? What's the deal? Why would you use sea urchin, right? So why would he use sea urchin? That's not a normal system. Who would do that? Okay. So generally, you can always ask yourself, and there's almost always a reason. Okay. The scientists choose these model organisms for some benefit, okay? And his benefit here has to do with the biology of. So we're going to the overhead again. Has to do with the biology of this. Let's say he was using humans or mice or something, right? And he needs to get fertilization to occur right away. His whole point is he needs a fertilization right away. And what if he's using human? Well, is he going to get fertilization right away? Okay, so here's the oocyte. And here's the sperm. You've seen these pictures, right? Where you've got like the human oocyte with all these little sperm trying to get in. You've seen them, right? It's pretty pathetic. They keep trying to get in. Everybody's trying to get in. They can't get in. Nobody can get in. Finally, one of them gets in, and then they create a thing so nobody else can get in. Okay, but you've seen these pictures, right? Why? Because that's just an extra quality control step that humans or mammals can throw in. They can make it hard. They can say, oh, we only want good sperm that can wiggle their way in. We don't want defective sperm, right? So it's an extra quality control mechanism. But yet, there's no way this is going to be synchronous. Okay. If you did this with several of them, they're all going to go in at different times because this thing is a rate-limiting step. If instead we're dealing with sea urchin are at the bottom of the ocean, well, or at least on the bottom of the, they don't have to be very deep, but they're down here. Let's say what's happening. Okay, so we've got they're underneath the water, and it's like particular nighttime, the temperature's right. It's the right season, and they're like, "Whoa, tonight we're gonna have children." And so then one of them is putting out oocytes, and the other one say, "Oh, I'm gonna put out sperm." Well, what's gonna happen? You got the ocean currents. It's not easy for them to find each other. So if they find each other, fertilization immediately. Okay, immediate fertilization. So what is Tim Hunt able to do? Tim Hunt is able to take a spoonful of oocytes. He's able to take a spoonful of sperm. He can mix them together, and he's going to get instant fertilization. Okay, why is that important? Okay, because he needs synchronous mitosis. So he needs them to fertilize at the same time and then go through synchronously through this. Okay, so now they're going through synchronously, and what he sees is that while he's got this one protein, ribonucleotide reductase, keeps increasing, with time. He's got other proteins that cycle. So they increase and then they go away. And then they increase and then they go away. And you're losing a little bit of synchrony. That's why doesn't look so good anymore. But when they go away is when they're going through mitosis. So he's got some proteins that cycle up, they get degraded in mitosis. Cycle up, get degraded in mitosis. And remember, it's just S-phase mitosis, okay? So it's relatively quick. And these are just time points, looking at what proteins are being made, because he's labeling with S35 methionine. And so he called these cyclins because they're cycling up and down. But he had no idea what they did, He just knew they were something that was being regulated by the cell cycle. And he figured they might be important, but he didn't really know. Okay, so the way this all came together was that one mutant that Paul Nurse, who did the fission yeast, was really interested in, CDC2, was found out that it bound to the cyclins. Okay, so that he had two cyclins, A and B. So it formed a physical complex with cyclin B. So this one is now called the cell cycle machinery, CDKs, which is a type of kinase. Remember, that's where they're adding on phosphate onto proteins. And they need cyclins for activity. It's called the cell cycle machinery. And then later MPF. So MPF really was late to the party because only later did they show that MPF, maturation promoting factor, or M-phase promoting factor, was CDC2 and cyclin B put together. Okay, so while they were sort of on the right track, they didn't have the tools to identify it. Okay, and so basically the people who were working on MPF did not get the Nobel Prize for that reason. But Paul Nurse, who did the fission yeast work, he got CDC2, which we're now going to call CDK1. Lee Hardwell, who did the budding yeast work, and he got CDC28, which is called CDK1. And then Tim Hunt, who did that sea urchin work, he got cyclins. So these guys are the ones who got the Nobel Prize in 2001. Okay, so now we're going to shift our focus, and we're going to talk about how these CDK cyclins are regulated, and then we'll go into how they're actually affecting these transitions. So it turns out that there's a lot of these CDKs and cyclins. It depends, right? So in budding yeast, where they did this work, there's only one CDK, okay? You can call it CDC28. In fission yeast, it's called CDC2, but you can just call it CDK1. It's the first one, okay? And at different points in the cell cycle, it's going to bind different cyclins, okay? And we'll go over some of these cyclins later. But there's G1 cyclins, G1S cyclins, S-phase cyclins, mitotic cyclins, and they're going to form active complexes at different points in the cell cycle. And then for vertebrates, we have a little bit more complexity. While we have different cyclins at different points in cell cycle, we also have different CDKs. So there's specialized CDKs for G1, G1S, S-phase, and mitosis. Okay? And we'll get into some of these later. So now we'll go over the regulation of these CDK cyclin complexes. So the one regulation is the CDK is not active by itself. So it has to bound a cyclin to be active. And then there's extra regulation. So there's CDK inhibitors called CKIs that could bind and either prevent the cyclin from binding or inhibit the whole complex. And so they're negative regulators. So they're stopping the regulation. And then there's phosphorylation of the CDK, and you can have inhibitory phosphorylation and activating phosphorylation. So we'll talk a little bit about this regulation. So I think we'll first talk about the inhibitory phosphorylation. Actually, we're not. We're going to talk about cyclins binding to CDKs. Okay, so the reason that they're going to bind different cyclins at different points is because the cyclins themselves are regulated, so they only show up at certain points. So you're going to have like this G1 cyclins like CLN1, 2, and CLN3, and they're only really at high level or even present at certain points in the cell cycle, and then they're degraded. Then you have S-phase cyclins, CLB3 and 4, mitotic cyclins, CLB1 and 2, and so the CDK can only bind them at certain points in the cell cycle. And we'll go over their degradation later. And then in yeast, in humans, we have the same kind of thing with different cyclins. They have slightly different patterns. Like you have like S-phase cyclin, cyclin A, mitotic cyclin, cyclin B. Cyclin D's is a G1 cyclin, cyclin E is a G1S cyclin. So basically, they're only around at certain points for the CDK to bind. And as we said before, there are different CDKs, and they're listed here. So like CDK4 and 6 are the G1 cyclins. Cyclin E is CDK2. It's for the G1 S-phase cyclin. Cyclin A can bind CDK1 or 2 because it's S-phase, and cyclin B is just mitosis, and we're CDK1. Okay, so now I think we're going to talk about this inhibitory phosphorylation. All right, so the way this works is pretty straightforward. So these are kinases. So they're going to bind ATP, and then they're going to take the last phosphate on the ATP and put it onto a substrate. Okay? So the way the inhibitory phosphorylation works is, a little bit hard to see here, but this is an ATP binding pocket. And what they're going to, the ATP is negatively charged. It's got 3 phosphates on it. And so the inhibitory phosphorylation is going to phosphorylate residues right in this binding pocket. And so because you got negative charges in the binding pocket, the ATP can't bind anymore. So it's just going to prevent the CDK when it's phosphorylated in the binding pocket from binding ATP. All right. And then there's activating phosphorylation, which is this T loop. Okay. And this T loop is, so here's the binding pocket. And in the activating phosphorylation, the substrate has to get here to be next to the ATP because they're going to take the last phosphate from ATP and put it onto the substrate. And the T loop is like a loop that's blocking it. And so substrate can't get in. But if this thing gets phosphorylated, then the T loop moves out of it just changes its conformation and moves out of there. And now the substrate can come in to that binding pocket. All right. So the inhibitory phosphorylation is going to be blocking ATP from binding. And the activating phosphorylation is going to get rid of this T loop that allows substrate to bind. So you guys shouldn't leave so early in case there's a pop quiz. Just letting you know. But do what you want. Okay. Maybe they have to leave for some reason. All right. Oh, we only have 2 minutes, right? All right. So, all right, let's do a pop quiz since there's like nobody here. Unfortunately, those people just left. That's sort of disappointing. You can lead horses to water or whatever.
What?
All right, let me turn this